Okay, so as I mentioned before, in a way, Mendel got very lucky studying the things that he did, the characters and traits that he did, because not everything is that straightforward, really. So the relationship between genotype and phenotype is rarely as simple as in the pea plant characters that he studied. Many heritable characters are not determined by just one gene with only two alleles. There's actually a lot of different other cases. But what he taught us has been like invaluable to figuring out, a lot of what does follow Mendelian genetics and then how it might deviate. So the basic principles of segregation and independent assortment apply even more, actually, to more complex patterns of inheritance. So there are complex genetics of a single gene, and we'll talk about how. And then there's also complex genetics of some things that are coded for by more than one gene. So we're going to talk about this first. So inheritance of characters by a single gene can deviate from simple Mendelian patterns in these situations. So if alleles are not completely dominant or recessive, if a gene has more than two possible alleles, and when a gene produces multiple phenotypes. So we're going to talk about a little bit about these in more detail. So what do you mean when alleles are not completely dominant or recessive? So these are the different cases that we're going to break down. Like those are the ways that it can deviate, and then there are some terms for different cases. So incomplete dominance and codominance is when the alleles are not completely dominant or recessive. Multiple alleles are when a gene has more than two alleles. And then when a gene produces multiple phenotypes, this is called pleiotropy. And so I will break this down on the next view, but I wanted to give this like overview so you can make headings in your notes. So the first case. So what we learned about with Mendelian genetics is called complete dominance. So one completely like masks or covers the other one. So like the purple flower color always overshadows the white flower color, right? So that is complete dominance that occurs when phenotypes of the heterozygote and dominant homozygote are identical, or homozygote, whatever. But there are differences. So in the case of incomplete dominance, the phenotype of the F1 hybrids is somewhere in the middle. Like it's a blend of the two parental varieties. And I'll show some examples. So instead of the flower color being either purple or white, if you cross these two true breeding parents, it's like if it was like a light purple. It's a blend. The phenotype is somewhere between the phenotypes of the two parents. And in the case of codominance, these two alleles are like both dominant and they're fighting to be expressed, and then they both get expressed at the same time. So in codominance, two dominant alleles affect a phenotype in separate, distinguishable ways. So in complete dominance, to show you an example, again, the phenotype of the hybrid is somewhere between the phenotypes of the two. So if you were to cross a red flower, this looks like a carnation, I would say, but I'm not really that good at it. Anyway, so if you crossed a red flower with a white flower, and then it ended up having a pink flower, that's different. That's not complete dominance. That is incomplete dominance. So one of them is dominant, but it's not like completely dominant. It ends up just being a blend. It's pink. So if you were to do a cross, it would be a blend. It's somewhere between these two different colors or the two different phenotypes of the parents. It's somewhere in between. Yeah, one is kind of dominant, but it's incompletely dominant. And that's opposed to codominance. So in this case, the two dominant alleles affect the phenotype, but in separate, distinguishable ways. So they both like are dominant and they're fighting for dominance, but they're like, well, I guess we'll both be dominant. They're co-dominant. And so in this example of like cow color, I guess this fur color is red, although I would still say it's brown. And then this fur color is white. In this case, they both each get expressed. So they're co-dominants. They're both like sharing dominance. They both get to be expressed. Nothing gets covered up. So they're co-dominant. They're like sharing dominance. So the two alleles affect the phenotype, but in separate but distinguishable ways. Like they're both present. And then I'll show another more complex example of codominance, but I think this is a nice one. So like if this flower instead ended up like splotchy red and white, then that would be codominance. But no, incomplete dominance is like somewhere in between. Neither one is dominant. Codominance, they're sharing dominance. And so again, it could be more complex. There might not just be this or that. There could be multiple alleles. So say in flower color, there was purple, there's white, there's also red, or there's some other color. Like there could be multiple alleles for one character or one thing. So most genes exist in populations in more than two allelic forms. And so kind of like our classic example is always the ABO blood group. So you know how you can be type A, type B, you could be type AB or type O blood, and that comes from multiple alleles. So there's actually 4 different phenotypes that are possible because of these different combinations. So the phenotypes of the ABO blood group are determined by 3 alleles for the enzyme that attaches A or B carbohydrates to red blood cells. So I'm going to show that example on the next slide. But these are the 3 different possible alleles. You can have IA, you can have IB, or you can just have I. So these are 3 different possible alleles, and then you can have 2, right? So wait a second. This is the wrong slide. Okay, I'm gonna have to come back to those slides. So the enzyme that is coded for by the IA allele adds the A carbohydrate to the surface of your red blood cells. The enzyme that's coded by the IB allele adds the B carbohydrate. So this could be on the surface of your red blood cells, or this allele adds neither. And so when we think about those combinations, because you're going to inherit one allele from your mom and one allele from your dad, it depends on what combination you get. So this is an example of, a red blood cell with only the AA carbohydrate on the surface. And so that means this person inherited either both copies of the IA allele or one IA and one I. That's how you get only the A carbohydrate. You can alternatively you could be type B, and so type B either got both copies of the IB or IB and I. So that is, an alternative method. And then this case, if you are type AB blood, then you have both being expressed. You have IA and you have IB. So this is actually the example of codominance because they're both dominant and they're both present. Both the carbohydrate A and carbohydrate B are present in their separate distinguishable ways. So this is also an example for IAIB. This is an example of codominance. But it's multiple alleles because there's another allele involved that could possibly end to another outcome. So if you happen to get both copies of I, then that's how you have no carbohydrates on the surface of your red blood cells and you are type O. So yeah, genetics are complex. So you can inherit different alleles and combinations of alleles, and that's what leads to different outcomes. And then let me go back to this. So that's multiple alleles. And again, there's like a relationship between dominance and phenotype. So whatever the dominant is leads to a structural difference. So for example, in P shape, the dominant allele is an enzyme that actually codes for like a different form of starch that's not branched, but changes it to a branched form. And so that's what makes it like smooth. And the recessive allele is for a defective form of enzyme, which leads to the unbranched starch. And so that's why it's wrinkled. So like, kind of like the flower color example with that one slide about the chromosomes, there's like something different that leads to these different phenotypes. Yeah. And so, for example, another example is this disease called Tay-Sachs disease. It is a fatal inherited disorder. And there's a dysfunctional enzyme that causes an accumulation of lipids in the brain. So again, you know, like whatever allele you get is actually making like a functional difference. And that's why this results in either a different phenotype or maybe a disease. And there are different degrees of dominance based on like what you're kind of focusing on, which makes it more confusing. So I think I'm just going to skip this part. And know that not all dominant alleles are going to be more common in populations. Just because in a probability way and in a Punnett square, it might seem like dominance always going to be more common, but it's not always the case in a whole population. It depends on what the trait is, and yeah. So, for example, one baby out of 400 in the United States is born with extra fingers or toes, and so this condition is called polydactyly, and this is actually a dominant allele. So it is technically dominant, but it's not that common, right? It's found much less frequently in the population than the recessive allele. Yeah, I think my cousin had polydactyly. So maybe his chances for his offspring are going to be higher, but that it's still not going to be higher in the whole population just because it's dominant. Okay, and so now coming back over here. So those were cases of one gene still, and one gene could have multiple alleles, but it could also be possible that one gene has multiple phenotypic effects. Because in reality, like when one thing causes a change, that could cause many changes. So for example, pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases, like cystic fibrosis and sickle cell disease. So these things cause a change, but it also causes multiple changes. It's not just only on one trait or one like character. It might have multiple effects. So it's genes having multiple phenotypic effects. Like if there was just one gene that was in charge of both eye color and hair color and whatever, if that was all one gene, yeah, or like sickle cell disease. So this one gene will have multiple different effects on the phenotype. Okay, so this is like a review summary of the complex genetics of a single gene and how they could be different from Mendelian genetics. So this is the simple Mendelian genetics, just complete dominance. Incomplete dominance is, again, when there's like a blend. The heterozygous phenotype is an intermediate between the two parents. codominance, where both phenotypes get expressed. Multiple alleles, that there could be multiple different possible alleles, not just two. And then pleiotropy is one gene having multiple effects on somebody or something. And so there can also be complex genetics of two or more genes. So again, when he was looking at two different things, two different genes, he got lucky because there could be these other super complex cases. So if he was studying these where it's two more genes, but it's more complicated, he would have just given up. Or maybe not, I don't know. So some traits can be determined by two or more genes. It's not always just one gene making one thing or another. It's usually more than two. So in epistasis, this is complex because it's when one gene affects whether or not another gene will get to work. So it's like a control switch over another gene. So in epistasis, expression of a gene at one locus alters the phenotypic expression of a gene at a second locus. And so the classic example for this case is in Labrador retrievers, the dogs. So in Labrador retrievers and many other mammals, actually, the coat color depends on two different genes. So there's one gene that we could say is B because it codes for the pigment color. And so that one determines which color it is. The other gene determines if there's a color or not. So one is like the on-off switch for the other one, basically. So one gene determines the color of the coat. If it's dominant, then it's black. If it's recessive, it's brown. So big B for black, little b for brown. So that's just coat color. And then the other gene is either dominant for color or recessive for no color. And that determines whether that color, whatever that is going to be deposited in the hair or not. So here is an example in visual. So I always thought golden retrievers were more common, but no, it's just common because we breed them that way. There are chocolate labs, and there are also black labs, which I feel like we don't see that often. I was really excited to see one the other day. So anyway, it's a tangent. So one allele or one gene, the B, is what color? If there's a big B, then that means it's going to be black. If there's a little b, then that means the fur color is going to be brown. And so anything that has a big B is going to be black. Anything that has both copies of the recessive are going to be brown. But if there is the recessive E, that means no color. And that's technically how you get the golden labs. The golden labs are technically no color pigmented pigment deposited into the fur. That's just kind of the color of the protein, I guess, how it looks. So if there's a capital E, then that means there's going to be a color. But if they're both recessive, then that means there's going to be no color. It doesn't even matter what the B is because it's just not going to get deposited. So the E is like the on-off switch for the color. So yeah, you can see it's a little bit more complex. So if two heterozygous black labs, and they're heterozygous for both things, if they're mated, you might expect the ratio to be 9:3 to 3:1, but it does not come out that way. And so they were like, what the heck, how? Like, what? It turns out if you do this, the Punnett square shows that it will be 9 black labs to 3 chocolate labs to 4 yellow labs because of this weirdness. So it's like these last two in the hypothetical 9:3 to 3:1 combined. So you still see the total number of odds. It's still going to kind of end up the same as like 9:3 to 3:1. But they just might be combined, like modified versions of this ratio. because, yeah, when you look at these combos, it should still add up to 16. And yeah, it'll just be modified versions of that ratio. But so again, epistasis is when one gene affects expression of another gene. And then lastly is polygenic inheritance. So in this case, it's just that it's even more complex, basically. So quantitative characters. So remember, quantitative stuff, quantitative data means like numbers. So things like measurements, height, things like that. Quantitative characters are those that vary in a population along a continuum. So like height is like actually a continuum. We're not just tall and short like these plants that Mendel was looking at. There's a continuum of different heights. And so quantitative variation usually indicates polygenic inheritance, meaning there's more genes involved. Poly means many as a prefix, so polygenic inheritance, like there's many genes influencing this one thing, like height. And so polygenic inheritance is an additive effect of two or more genes on one phenotype. So height is a good example. You know, we're all a range of heights. It's not just your 5 feet or your 6 feet. There's a range, and that's because there's over 180 genes that affect height. So all of these different genes affect this one thing, this one phenotype. And skin pigmentation in humans is also controlled by many separately inherited genes. So here's kind of an example. This is like simplified, but this kind of shows the range of different skin colors. Although this is like the foundation island, right? Like it's really not that inclusive. But there are multiple genes that can be involved in like different continuums on a trait. So those are complex genetics of two or more genes. Epistasis, when one gene controls another gene being on or off. And then polygenic inheritance is like there's multiple genes involved in this one character, this one phenotype. And then there's also other things too, like nature versus nurture. There are some things you inherit, but there's also some things that you can change about like your own life. So another departure from simple Mendelian genetics is when the phenotype for a character depends on environment as well as genotype. So the phenotypic range is like broadest for polygenic characters. Traits that depend on multiple genes combined with multiple environmental influences are called multifactorial. Yeah. So here's an example. Hydrangeas, they can change their phenotype based on environmental changes. So the pink versus blue isn't actually like different alleles. The same hydrangea can be either color based on the acidity of the soil. I was watching that show Bones. It's kind of older now. It's like this lady who's like a forensic, does forensics, whatever. And so it was like, oh, somebody must have died here because blood is a little bit acidic. And so this changed the color of the hydrangeas that were growing nearby. Yeah, like, so these different colors are not due to totally different alleles. It's that this same species and type of hydrangea grown in basic soil, if it was the same thing grown in acidic soil, then it would change the phenotype and be a totally different color. So that's how environment can also affect phenotype. And then an organism's phenotype includes all aspects of its physical appearance, internal anatomy, physiology, and behavior. Like there's just a lot that actually does go into phenotypes. So for example, if you need glasses because you strain your eyes a lot, doing something, or like that was an environmental effect based on affecting your phenotype, not just like whether your parents have glasses or need glasses or not. You know, there's a lot of stuff that affects our phenotypes. So in organisms, phenotype reflects its overall genotype and unique environmental history as well. Okay, I'm gonna have to make a part 4.